We're going to talk about how epinephrine, which is a small molecule that is produced in your adrenal glands, which sit on top of your kidneys, and then released into your bloodstream, stimulates liver cells to convert glycogen, which is a starch, to glucose. So let's get started. So epinephrine is a small molecule, and it stimulates protein receptors that are on membranes. And so a molecule that stimulates protein receptors is called a ligand. And then molecules that are produced in one area of the body and transferred and then stimulate another area of the body are called hormones. So epinephrine looks like this. So if we look at the structure of epinephrine, you can see that it has a nitrogen group over here and then these hydroxyl groups. And so it's because of this nitrogen and hydroxyls, this molecule is soluble in water. So we say that it is polar or hydrophilic. And because of that, it will not cross a cell membrane because the center of a cell membrane is a lipid, which is nonpolar and hydrophobic. So let's look at how this epinephrine molecule will get into a cell. So from now on, epinephrine will be a green dot in my drawings. So here we have a cell membrane, and we have to get this green dot, epinephrine, into the cell, or we have to get the green dot to trigger a response inside of the cell so that the cell knows to make glucose. So the other thing I didn't tell you about epinephrine is it's part of the fight or flight response. And so if you see something scary or you hear something scary, if it's imagined or perceived, your body will cause a response of epinephrine into the bloodstream, which then will change your physiology to be prepared for a fight or to be prepared to run. And so this molecule has to tell the liver cells to produce glucose because the muscles are going to need this glucose in order to run, because glucose is energy. So it's going to trigger a receptor site on a membrane-bound protein. And this membrane-bound protein here is an adrenergic receptor. Which this basically means is going to get a stimulus and trigger something else. This particular adrenergic receptor is going to stimulate a G protein complex. And so G proteins are a 3 subunit structure like this. Okay, 2 of them are slightly bound inside of the membrane here, and then it has a 3rd subunit. And these have names like alpha, beta, and gamma. And that's an alpha sign. And what's going to happen is that on this alpha is a GDP molecule, which is similar to ADP. So if you remember, ATP is our high energy molecule, which drives a lot of reactions by adding a phosphate to them to make ADP. And so this is adenosine triphosphate, and this is adenosine diphosphate. And then this P is a phosphate. So GDP is, instead of adenine being the nucleotide that makes up this structure, it's guanine. And so you can have GTP, you can have GDP, or GMP. Okay, so these things all just have different amounts of phosphates, 3, 2, and 1 phosphate attached to them. So this alpha subunit of this G protein has a GDP attached to it. All right, so now what's going to happen is that this epinephrine molecule is going to move into the binding site. It's chemically attracted to it, and it's going to then sit right here in this binding site like that. And when it does that, the tail over here will change slightly on a molecular level, and that's going to then change the response of this G-coupled protein. And it will break apart this alpha subunit from the beta and the gamma subunit, so that this, and I should say, GTP will come in here and replace the GDP. So GDP gets replaced by GTP, and the alpha subunit will move away from the beta and gamma subunits. So we have now our alpha subunit here, and it has a GTP on it. So and then we have GDP in solution. Okay, so these G proteins, these green guys here, because it's being activated by this adrenergic receptor, we can call this a G protein complex receptor. So G protein complex receptor, or GPCR. So now what happens? So our goal here is, remember, we're trying to make glucose. And what's going to happen is this alpha GTP complex is going to stimulate, you guessed it, another protein. Okay, so it's going to trigger a response in another membrane-bound protein, adenylyl cyclase. So this has some subunits in like that and some subunits in like that, and then its structure kind of hanging out down here. So our GTP migrates over to this structure and then eventually gets in contact with it. Okay, so we have this migration happening, and then it touches there. Now when the GTP, G protein GTP complex touches our adenylyl cyclase, so adenylyl cyclase, what that's going to stimulate then is the conversion of an adenosine triphosphate molecule to a cyclic adenosine monophosphate molecule. So this is adenosine, which is the base, triphosphate. And then this is cyclic adenosine monophosphates. So while I'm writing this out, how many phosphate groups are on there? So cyclic adenosine monophosphate looks like this. And so the conversion here, is taking this ATP molecule that I'm going to write in red. So it has 3 phosphates, 1, 2, 3. Here's the ribose, which is a sugar. And then here's the adenine base. And so it has adenine triphosphate. They all have a ribose sugar here. This one then, what happens is 2 phosphates come off, 1, 2, and you end up with adenosine monophosphate. But you see how this is in a cyclic pattern? So that means cyclic AMP. And so that's what's happening when this G protein touches then adenylyl cyclase to convert ATP to cyclic AMP. So you might be asking yourself, what is the point of all of this? The point of all of this is that cyclic AMP is a secondary messenger. And it's going to trigger then responses on other protein molecules inside of the cell to tell the cell to start cleaving glucose molecules from glycogen. So how does that happen? Well, cyclic AMP is going to stimulate protein kinase. So the protein kinase has 4 subunits in this example. And it has 2 of the subunits have active sites that are blocked when the regulatory sites are connected. So these are our catalytic sites and our regulatory sites. So regulatory in red, catalytic in green. So cyclic AMP, that small molecule, is going to be converted over and over and over again. So one epinephrine molecule will convert a lot of cyclic AMP because this thing stays attached. So this process just keeps going. It doesn't stop. It just amplifies the manufacturing of cyclic AMP. So you end up with a lot of these cyclic AMP molecules floating around, and then they start to bind into these protein sites. And when that happens, this molecule will then split apart and release the active sites here to do some kind of work. So we end up with then our regulatory sites and our catalytic sites. So the catalytic sites now are free to do work, to do something. And the type of work they're going to do, because I told you this is a protein kinase, kinase just means that they're going to phosphorylate something using ATP. So ATP becomes the carrier of the phosphate, but it's the protein kinase that will actually stick the phosphate onto something. And the something that we want to stick the phosphate onto will be this protein complex over here called, is going to be stimulated by protein kinase, and ATP will come in so that this gets phosphorylated. And so when this molecule is in the active state, now that it's phosphorylated, it's going to take a molecule called glycogen. And glycogen is made up of many glucose. Each one of these circles is a glucose molecule that are linked together by their alpha 1,4 carbon bonds. And so this molecule here, glycogen, will bind with our glycogen phosphorylase, like that. And this then will release one of these, I'm going to make this in red, glucose molecules, and we end up with a glucose 1 phosphate. What we're going to do then is take this molecule and it's going to get shuttled out of the cell. Okay, so this molecule then will eventually be converted to glucose and then be transported out of the cell using other proteins. So there's a lot of other steps going on here before it gets transported out. But one thing I want to point out is that you amplify the CMPs here so that you get lots of them. So you're triggering many of these protein kinases, not just one, but you're triggering thousands of them. And then the thousands of protein kinases are turning on lots of glycogen phosphorylase. Okay, I think I wrote phosphate here, but I meant glycogen phosphorylase. which then cleaves a lot of glucose molecules. And so you get about 10,000 glucose molecules produced for every 1 epinephrine molecule stimulation. So this is an amplification effect, right? So this whole thing is really just being amplified. Through each one of these cascading steps.